Okay, thanks very much. So yes, Ram Kumar, and I've been asked today to talk about, let's see if this works, about the emerging treatments, or at least to introduce the topic to you. So this is, I'm not going to go into the individual treatments in too much detail because there's so much vast amount of experimental treatments that are coming along, and the specific companies are talking about their treatments that are in the pipeline afterwards. So I'm just talking about general principles mostly. So I'm based up in the northwest. I do some work in Manchester, but mostly up in Preston at the moment. Less so in Liverpool, and I do some work at the Evelina in London with Dan, who's great contact. So disclosures, I have undertaken paid consulting for Acadia, who's one of the pharmaceutical companies who's presenting here as well, and I'm a partner in CYP Neuro, which is essentially an educational resource in paediatric neurology. Loss making. So, summary. So what I'm talking about is an overview and reminder of Rett syndrome, as if you need a reminder, but specifically where it relates to treatments, and just broadly going through the types of treatments, current treatments that are closer to the market, we can say, and then emerging disease-modifying treatments, and then something about the journey from lab to pharmacy and what you may need to know as somebody who may be asked to participate in trials, or even if the trials are finished, how do you get hold of this medication? And then some of the challenges and considerations that apply to all of this. And I'm speaking partly because I've been a principal investigator in clinical trials, one in Rett syndrome, as I was saying, with medication, and also in other conditions as well. So I can see where some of the challenges are coming up. So Rett syndrome, we heard chapter and verse earlier today, but it's always worth reminding oneself. So in essence, classical Rett syndrome, the MECP2 gene mutation, that's the key cornerstone of the condition. It's essentially the instruction manual to this protein that's damaged in Rett syndrome, and it's an X-linked condition, meaning that it's typically girls who are affected and women, but there are some individuals who, for unusual reasons, it's male, and then there's a related condition, genomically related condition, where you can get too much MECP2 as well, and that affects boys with a somewhat different condition, but similarly very neurologically disabling. And that is important when it comes to these disease-modifying treatments. So in Rett syndrome, this MECP2 protein and gene is dysfunctional and generally has reduced function, hypofunctional, or no function. And as we heard earlier, the MECP2 controls the production of other genes. It's involved in switching on and off other genes. So it acts like a volume control on a lot of other very important proteins that you need for brain functioning and functioning elsewhere in the body. And it's important for brain development and also maintenance as well, maintenance of the nerve cells. And it controls particularly the synapses, the connections between brain cells, and how they connect to each other, communicating through electricity and neurochemicals. And I'll put it, it's somewhat like Goldilocks, you can't have too much when you can't have too little of the MECP2. It has to be just right, because either can cause problems, and that is one of the challenges with these gene therapies, where you're trying to replace this hypofunctional or no function protein, that if you have too much of it, that can cause issues as well. So there's a concept of disease-modifying treatments, and when it's applied to a situation of Rett syndrome, typically what people mean by disease-modifying treatments is something that's very close to a treatment that's trying to get close to the root of the problem, near the MECP2 gene, either replacing the MECP2 gene or a protein that's close to it downstream of this genetic problem. We're trying to get around the problem. And the great hope is that you're trying to reverse the condition. I mean, that's what you'd really want to go for. And rather than managing the symptoms, which is what we have to do at the moment. So the long-term effects of those treatments could affect the way the disease changes over time. And we heard some information about how the condition can appear at below age 5 to 10, 10 to 15, and into adulthood. So the examples of these disease-modifying treatments are gene therapies, but more generally there's other types of treatments like stem cell treatments you might have heard of in other conditions where you're trying to replace certain cells that might be particularly important. Gene editing you may have heard about in the news. It's been mentioned in other conditions like sickle cell disease where that can cure the condition, at least in some individuals, and that's where you're trying to repair the gene as well. So these are some of the disease-modifying treatments. And then symptomatic treatments, that's in terms of managing individual symptoms. So you're managing the respiratory problems with antibiotics, let's say, or managing the tone with baclofen, for example, or botulinum toxin injections. And the idea is that you're trying to improve disability or improve quality of life, pain, these types of symptoms. And generally, they don't change the underlying problem. So the underlying condition continues on its trajectory through childhood and adulthood. And if you stop the treatment, the symptoms come back. So just as a reminder, through the current symptomatic treatments that we've got, which are I've concentrated on the medications, but there are non-medication treatments as well, as I indicate there. So seizure management is the classic from our point of view as paediatric neurologists, with anti-epileptic medications, and there's non-medication treatments that we had some discussion about ketogenic diet as well, and vagal nerve stimulator. Tone management, another area where we're quite commonly involved as paediatric neurologists. So dystonia, which is abnormal tone and sustained posturing or excessive movements. Spasticity, which is a type of muscle stiffness that can be seen in some individuals with Rett syndrome. Respiratory problems, which we'll hear more about tomorrow. Feeding, so there's a significant gastrointestinal problem, so there may be treatments such as antispasmodics for the gut, for example, or anti-reflux medications. Sleep management, so significant problems, so a number of individuals may be on melatonin or other sleep agents. And then a large list of medications based around anxiety and the behavioural components, as Professor Santosh was talking about. And then some of these more therapy-led interventions, which we can broadly call rehabilitation. So maintaining mobility of function, occupational therapy, speech-language therapy, and we had a discussion about nutritional support and dietitian as well. That's obviously very important. So, as I say, the limitations of the current treatments that, in the perspective of disease-modifying treatments, as I mentioned earlier, it doesn't directly affect that lack of the MECP2 gene and its product, the protein. So most of these treatments that I've mentioned, the symptomatic treatments, are taken from other conditions, and you've borrowed them, expecting them to work in a similar way in individuals with Rett syndrome. But there are some specific issues. As we were hearing, there's a lot of autonomic disturbance. There's an element of long QT syndrome as well that can affect some of the other medications. There's the respiratory problems that can interact with the tone medications that tend to make your respiratory problems worse sometimes. And as I say, some of the progressive problems like scoliosis, these orthopedic issues may continue unabated. So that's the reasoning for having disease-modifying approaches. So it's not an either-or, because even with disease-modifying treatments and the most advanced gene therapy, there's still going to be a need, realistically, for symptomatic treatment of some form affecting some part of the body. And I've put in there role model is spinal muscular atrophy. So you may have heard of spinal muscular atrophy. It's a very different condition from Rett syndrome. It affects the nerves to the muscle, and muscle function can cause significant physical disability and scoliosis, as we see in individuals with Rett syndrome, but doesn't tend to affect brain function, cognitive function. And this is a condition which I find useful to think about. It's gone on that journey from symptomatic treatment to disease-modifying treatments in the space of the last 8 years. And it's really changed the nature of the condition, but it's brought its own challenges as well. So that's why I say, it's not a case of disease-modifying treatment, and we don't need to think about symptomatic treatments anymore. So gene therapy, which we'll hear about in more detail from the company representatives, it essentially works at its simplest level by trying to put a working copy of the MECP2 gene back into the brain and getting it into the brain. And the idea behind it, or how it's labelled, is it's a one-time treatment. Now, that means, I've put for better or worse, it's a one-time treatment. I mean, that's just pretty much a statement that it's a treatment that you get once, or that's how it's visualised, but it may not necessarily remain functional for a lifelong period, and that's one of the reasons why the trials have an extended period of observation. So the challenge is to get the gene, a working copy of the gene, back into the brain, and your body protects your brain quite thoroughly. You have something called the blood-brain barrier, which tends to keep out a lot of agents. So, and that's true for any toxins in your bloodstream and infections as well. But in this case, you use a virus essentially like a Trojan horse. There's a particular virus called the AAV9. It's an adenovirus. It often causes a cold or gastroenteritis. But it's a specially modified version of this virus, and it's specially recombinantly engineered so that it carries a copy of the gene back into the brain, and hopefully the body accepts this transplanted gene and assumes it's part of its own genetic makeup and starts producing the protein. And that theory works in spinal muscular atrophy, and it works in a very sustained way. So that's given a lot of hope in terms of actually this is a viable prospect. So the idea is that this virus, the Trojan horse, will get into the brain, and the MECP2 starts producing proteins, but it has to be just the right amount, as I say. So it's the Goldilocks situation. You can't have too much of it, and you can't have too little, and that's somewhat different from the other condition I mentioned, spinal muscular atrophy. So that's what introduces some of the additional biochemical challenges. So the gene therapy has to get into the brain, and I say that if you have the blood-brain barrier, which prevents it getting in for good reasons, so it has to be delivered typically through a lumbar puncture, so that we call that intrathecally, or into the ventricles of the brain, so introduced through the skull into the spinal fluid that bathes the brain. So that's generally what happens at the moment, and so there are other conditions where you can give the virus intravenously through the vein, but it appears in this condition to get the right amount of the gene working copy and the virus into the brain, you need to give it through one of these routes. So there is an invasive component to it. And I say there's a particular virus that seems to be latched onto, and there's multiple strains of this virus, and it's modified in different ways. But one of the issues is this virus can cause disease. It can cause hepatitis particularly, and it can affect blood cells as well, risk of bleeding. So that's what introduces some of the safety challenges with using this virus, and you have to give a particular high amount, large dosage, much higher than you'd get in a natural infection. So that's why there's some caution regarding the side effects of that particular virus, which are potentially life-threatening, and that's certainly been seen from the experience with spinal muscular atrophy. So the MECP2 gene is modified, and each company will have its own particular version of modification. So sometimes it might not be the whole gene, it might be a portion of the gene that's felt to be the most functional, or it's modified to prevent overexpression, so getting too much of the gene as well. So the companies are currently, they're talking at this conference about the current human trials, and I say the safety and efficacy is one of the key indicators that you have to look at. So is it safe? So is it causing these problems with liver function, blood function, bleeding, etc.? And does it actually work, which might take longer to work out. So just in terms of the path to get to the human studies, there's a long pipeline even before you get to human studies. So I remember reading about the work at Edinburgh, some 20 years ago or nearly that, and naively I assumed, okay, well, this is mice or rats, so it's maybe 5 years, possibly 7 years, not more than 10 years. To get to actual treatment, and that's extremely naive. So there's a pipeline that starts in animal models and cells, looking at, can the underlying treatment potentially work, but it's not in a state that you can actually use this in human beings. So there's work that showed that it can extend the lifespan of mice with a particular form of Rett syndrome, an engineered form of Rett syndrome. It can improve motor function as well, and it can improve brain tissue function as measured by the electrical EEG patterns. So those models are still necessary, and people are still working on it. I've done some literature searches just to see what people are working on. So people are working on non-virus methods of getting the gene into the brain cells, so you don't have that issue of the potentially serious illness that the virus can cause. So, even though that there are trials happening now, there's still further advances in the pipeline which come to fruition over the course of years. So you have to think of things in what's likely in the next 2 years, what's next likely in the next 5 years, 10 years, etc. And human studies, of which there are much fewer for disease-modifying treatments, but there's many more for more symptomatic treatments, are they're split into different phases. So you might see talk of phase 1 trials and phase 2 trials, and they'll have different inclusion and exclusion criteria. Some might just be children. Some might include adults as well, and there's reasons for that. So particularly with this virus, it seems the effectiveness is related to the dose, and the dose is based on your body weight, but also the side effects are related to the amount of virus you're given. So if you've got a higher body weight, that can mean potentially that you're getting a larger load of virus and significant effects from that. So that's partly drawn from experience from SMA, spinal muscular atrophy. And with the human studies that are being undertaken or are being planned, the AAV9 is used to carry this MECP2 into the spinal fluid, and we've got Taysha and Neurogene. But there are other companies and countries that are potentially involved. So I found one trial that's initiating in China, I think, and the place to find these trials at various stages of setup is in a couple of websites. So there's one called Clinicaltrials.gov, and another one called bepartofresearch.nihr.ac.uk. So they're searchable databases which have got these trials registered, and Clinicaltrials.gov is based in America, but it's international. So the Chinese study, for example, is on the Clinicaltrials.gov website. And the nihr.ac.uk is a UK based, as it suggests. We've got Acadia here to talk about this particular treatment called trofinetide. So this isn't a gene therapy; it's downstream, somewhere downstream of the gene itself, and one can call it disease-modifying, or one can call it symptomatic treatment, and it's already approved in some world regions, as well here, but not so far in the UK. And essentially it's an oral medication which is a liquid, and it modulates the brain chemistry, in essence, somewhere downstream of the MECP2, and we can hear more about exactly how that works. But the clinical trials results, and this is the difference between the gene therapy trials and trofinetide, is it's studied in what's called a phase 3 study. So phase 3 means it's been studied in somewhat larger groups of individuals, usually with a placebo, meaning a medication that doesn't work but looks like the active medication. They found a statistically significant difference, perhaps clinically significant. We can discuss what that means later as well. But there are other similar products in development or trial or planned trials as well. I say the pipeline can be several years. Now, there's a whole vast area of pipeline products, these emerging technologies which are based around the molecular targets. Now, so this is a very broad brush review, but I had a look through, did some searching and some reading about what people are working on. And you may have come across RNA, which is different from DNA. It's one step down from DNA. It's basically like the message produced from DNA before it gets to protein. And you may have come across that from the COVID vaccines. That's based on RNA technology. And there's a whole area of work on RNA-based treatments. And once again, SMA, spinal muscular atrophy, is the role model for this, because it's now used in clinical practice. It's pretty much routine, available and expected that you receive one of these treatments in spinal muscular atrophy. But that was only from about, I think, 8 years ago. And it just seemed to come out of the blue, with no warning. But obviously people have been working on it quietly in the laboratories for many years, but it seemed to come out of the blue. So you may have heard of antisense oligonucleotides, ASOs. So I couldn't find anything specifically in Rett syndrome, but there is a US trial in MECP2 duplication. So in that situation, the MECP2, like I said about the Goldilocks situation, when you have too much of the MECP2, it also causes problems. So the antisense oligonucleotide there reduces the amount of MECP2. But in theory, you can have antisense oligonucleotides that increase the level of the MECP2 product as well. And then there's other molecules that work on RNA that don't count as antisense oligonucleotides. And the difference here is, so the antisense oligonucleotides, at least the ones I know about, go through the spinal fluid again, and they don't have a lifelong effect. So in the example of spinal muscular atrophy, it has to be given repeatedly as well to maintain its effect. So you need repeated lumbar punctures in that situation. Now, the other small molecules that work on RNA in the role model condition, spinal muscular atrophy, that's given orally. So it's an oral medication, it's given daily. So you've certainly got, you could say convenience, but obviously it does involve reliably taking medications daily, which can be a challenge in individuals. And we see that even with the symptomatic treatment. So that's why people continue on what appears to be a more cumbersome method of delivery of treatment, even when you say, oh, look, there's an oral medication. So that's, I think SMA is quite a good role model because it's gone down that pipeline in the last 8 years. And then there's something called RNA editing, which is, you may have heard in the news about, there's a technology called CRISPR, and there's base editing as well, which I think there was a particular scenario, and I think it was an immunodeficiency or a tumour condition in a child that was treated at, I think it was Great Ormond Street recently, with these technologies. So these technologies are coming in for editing, changing either the MECP2 itself or the RNA downstream of it. And there's a trial in China in MECP2 duplication, but I haven't seen anything in Rett syndrome yet. So, but it should be amenable. It's not a very big gene, it's not a very big protein, so it should be amenable to that. And then genome editing, I say that's getting more directly to the problem itself, but it might be a bit more difficult than RNA treatments. So trying to fix the gene mutation. We heard something about the X inactivation that you get in females. This can explain why some females aren't affected as much as you'd think, because they've got what they call skewed inactivation. So they're producing much more of their normal copy of the MECP2. So there are technologies that try and reactivate the normal copy and suppress the abnormal copy. But these are very much in the early research stages. And what we've seen with SMA, once again, the role model condition, is individuals might be on multiple types of treatments at different times. And watching some of their specialty group webinars, there's extremely detailed discussions about what do you do with a 25-year-old with SMA who's got scoliosis and you can no longer do lumbar punctures for ASO. So I think some of these situations will come up in the future. So just going back to the human trials, and just an idea of, because you'll see this sometimes if you look on these websites, it says phase 1 or phase 1/2, phase 3 trials. What does that mean? Because you may be asked to partake in it or sign your child up to partake. So what does that actually mean? So is it phase 1 usually means, it could be first in human in essence, or first in small number of participants. And normally in most conditions like hypertension, let's say, very common conditions, it's usually done in normal volunteers first. But when you've got rare condition like Rett syndrome, often this is done in individuals with Rett syndrome. So you may be asked to participate in quite an early stage trial. And phase 1 is really not looking at whether it works. You may hope that it works, and the people who are doing the study might hope it works, but actually it may not work. And what they're looking for is signals like what's the most you can tolerate, what's the most virus or virus agent, similar agent that you can tolerate. So the signal safety on that, and looking for side effects rather than looking for positive effects. And I think that's important because you may be excluded from other trials which may work because you've participated in this trial. So there's an element of that. They're often called phase 1/2 trials that I've seen. And I say that's probably not, so that in a rare condition you're not subjecting people to things that don't work at all. So it often overlaps with phase 1, and that's looking at, does it work in terms of certain target symptoms that you have, or other indicators like, what's the head circumference doing? Do you see a fall off in the head circumference centile? Is there a change in the EEG, for example? That can give you a clue that something's working at a brain level. So there'll be a number of different outcome measures there, either the ones that we discussed earlier, or biomarkers, which we can discuss what those are. And I say that they're still looking at safety, because that is the big issue here, particularly using this adenovirus in gene therapies. And you may compare it to placebo, or it may be unconcealed, meaning you know exactly what you're getting, and the issue is you're giving different doses to see what the effect is. So phase 3 is where you're getting closer to saying, okay, we want this prescribable from the pharmacy. So you've got a much better idea that this is a treatment that is going to work, but you need to get evidence together to convince the regulatory authorities and pharmacies, the hospitals, that you should prescribe this because it does actually work. So they tend to be larger studies. Now, this isn't always true in rare conditions. In rare conditions, sometimes they call them orphan drugs, and they give a bit more leeway to the amount of evidence that you need to get medications actually onto pharmacy prescribing. But in general, they tend to be multi-centre, so not just one centre. Maybe if it's a rare condition, it may be 2 or 3 or 4, 5, 10. And they tend to be often international, because to get those kind of numbers in a short space of time, you might need to get a number of centres internationally, rather than wait for 5 years to get the required numbers. And there's a lot of statistics in how to get the right numbers to show the right effect. And this phase 3 trial provides the strongest evidence that the treatment works and is safe. It's not... You still can't say it's 100% safe, because sometimes you have to follow people up for 4, 5, 10 years and have it available on the pharmacy before people go, actually, it wasn't as safe as we thought, and then it gets taken off the market. And we're very used to that in clinical practice. So at this stage, it's usually compared to placebo, and that's worth knowing about. Because if you're recruited to a phase 3 trial, then you may not actually get the treatment. You may get the placebo. But typically they'll design the study so that everyone gets a chance to get the treatment after a lag, basically. So that's what the open label extension is. So everyone gets a chance to go get the treatment. And it's also used as a way of showing long-term effects as well, because usually trials, you can't necessarily follow up people for decades. You need a shorter window of time that you're getting people back or imposing on their everyday life. And it's also burdensome for the people doing the study as well. So it's trying to get a balance between getting the required amount of data in a shorter space of time as possible, but across the relevant outcomes. So how do they measure outcomes? And this is important because certainly in the phase 3 study, even earlier, you have to say that it actually works, to take it forward to the next step. And in the phase 3 study, you need to show that it's causing a demonstrable improvement, often in terms of not just seeing a different symptom, but also in terms of function, reduced care needs, reduced healthcare costs. So that's a tall order, actually, a very tall order for a trial to do. So, but some of the traditional measures, you'll come across, or you may have come across the Rett Syndrome Behaviour Questionnaire, RSBQ, that's often used across a number of different studies, for better or for worse. It's got its own issues as well, but it's a questionnaire that you fill out, and that can tell you about whether the treatment's working, at least on behaviour. There's something that we already mentioned earlier, the clinical global impression scales, which is basically somewhat more standardised, but in essence it's saying, do I think they look better? So a bit more standardised than that, but it's got its problems. And then there's other scales that we heard about with Professor Santosh, I think, talking about another scale that captures the multiplicity of effects of Rett syndrome, motor function measures, and that, once again, coming back to safety, adverse events as well. So the limitations are, subjective interpretation by carers, and by the clinicians. You hope it's working, so you may go, I think this my clinical global impression is somewhat better than the next level down, because you're hopeful. But you also may miss subtle improvements that only become evident over years. So if you have to assess somebody at 3 months after the treatment, actually the full effect may not be seen for a year. So you may underrate the improvement at 3 months. And it doesn't capture all aspects of the condition. It may not capture the autonomic dysfunction, for example, or changes in long QT, etc., or scoliosis. So there's a number of different approaches. So physiological measures, we heard about heart rate variability, breathing patterns, wearable sensors, movement tracking, brain imaging, and blood biomarkers. These are all done in other conditions where you think there's something happening in the body that you can't necessarily detect on the outside. So there's a number of those being developed. And then this is from my knowledge of interacting with these. I mean, we're still not sure who holds the money, the purse, sometimes. But once you've shown that the treatment works in these trials, that doesn't mean you can just go ahead and prescribe it. It has to go through several hoops, and this is based mostly around England. I think Scotland's got its own version of things, but it has to go through the MHRA. They actually say it can go ahead to a trial in the first place and do the safety monitoring. But then you've heard of NICE and you've heard of NHS England, which is going to change again in the near future. So they assess, is it cost effective? And they might have their own particular thresholds of, okay, we don't think it's cost effective enough. And you may argue with that, and that often comes up. The family groups will often argue about, actually, you've said it's not effective, but you haven't taken into account this such and such, parental care, non-paid care, etc. And they make recommendations for NHS funding or not. And NHS England, which holds the money, will say, okay, this is such an amazing treatment and has such an impact cost-wise, the minimum, in the long term, that will pay for it. So once again, the role model is SMA, spinal muscular atrophy. So you may have heard it costs, I think they got a deal, and the deal was, I think, £1.8 million, for the treatment. So it's the most expensive drug, we can say. But the effect of it is so amazing that it's felt to save that much money in a not very long space of time, just in terms of healthcare needs, let alone anything else. So that's the kind of hoops that people have to jump through, and you may have to collect data prospectively to get that kind of information. We've got people talking about that today as well. So realistic timelines. So what's available now? So trofinetide, where we've got Acadia talking. As I say, it's improved in some regions, but not in the UK at present. The enhanced supportive care, the symptomatic treatments are available right now. Next 2 to 3 years, the gene therapy trials are already ongoing elsewhere. So they'll start reporting results from current trials, and also the safety data as well. And once again, depending on how effective it is, they may rush things through and say, okay, we don't need to do further trials. This is enough, actually. And now it's become a regular treatment. There's certain hoops you've got to jump through. Even after that, you have to prepare individual trusts to actually deliver it. But they've done it with SMA, so done it very rapidly in SMA. So in the next 5 to 10 years, there's some of those other treatments that I mentioned that are still mouse models, let's say, which are likely to come into the human childhood and adulthood trials. And combination therapies will likely get tested as well, and more targeted approaches based on specific mutations. We saw a slide about certain mutations seem to be less severe and others may be more severe, bit of a range even within that. But certainly we see targeted treatments in certain other conditions like Duchenne muscular dystrophy. That's another muscle disease where there's targeted treatments at particular types of mutations. So I think really, there's obviously a lot of things going on, but there are some expectations. Safety is the top priority. There is a treatment effect and side effects, and they can take years to become clear, which sometimes means you don't see anything very quickly. The study results don't come out very quickly, or you may be enrolled in the study a lot longer than you'd like, with potentially repeated assessments. Treatment efficacy, not every treatment will be effective that you may sign up to. And then individual responses, may be different effectiveness in different individuals, even given the same treatment. And also, the proven treatments, the symptomatic treatments, whether it's the therapies, for example, you don't need to neglect those because there's a newer treatment that's come along. Then, I say the regulators can take time to make decisions, and they may not necessarily come up with the decision that you'd like as well. And as I say, the health economics of it may not work out, and we've seen that with a lot of other medications that have been in the news. For example, in Alzheimer's with the monoclonal antibodies. So, what to consider in a trial participation? So it's essentially who, what, where, how. And recognise that your child may not meet the inclusion-exclusion criteria of the study. It may be related to a certain material of the gastrostomy. It can't accept that particular type of liquid that they've used for the medication, or the severity of other illnesses. There may be too much scoliosis or respiratory problems at this stage to be able to have somebody who's unlikely to to exit the trial early, for example, because of side effects. Travel requirements, it may be that you have to, in some of the gene therapies, you may need to be an inpatient because of delayed response to the delayed adverse response to the virus, and intensive monitoring periods, and say placebo, so individuals may get placebo rather than the active drug. And then some commitment to the beyond the main trial, so some element of follow-up over the 5 to 10 years afterwards. So some of the questions that you may want to ask the companies or trialists: what stage of the development process? What's the prior evidence of effectiveness? What's the size of effect, we can call it. The durability of effects, although we call it one-time treatment, not all these genes remain functional in the brain. Certainly in SMA it appears to remain functional, but these are different cells they're targeting. They're targeting cells a bit further down than in the brain. And what are the main side effects of the treatment? And then, potential start date for UK trials, which may lag based on the original optimistic ideas. Eligibility criteria, so look at what the inclusion-exclusion is often by age, but it will often be by comorbidities, and it may be based around individual mutations as well. Physical site versus remote visits. Since COVID, there's been a move to get more remote assessments undertaken. And what's the data measurement burden in terms of filling out a whole load of forms or having blood tests and other potentially invasive tests being undertaken? How long does it last? What's the follow-up provided? And also specifically, let's say the medication is found to be effective. How do you maintain the medication, if it is medication, after the trial finishes? So some pre-agreement about that. And then, what is the plan about gaining NHS approval? Because you may get to the end of the trial and then actually, the NHS isn't going to necessarily accept it unless there's evidence that it has a significant health economic outcome. That's some of the clinical trial information. So Clinicaltrials.gov, be part of research.nihr.ac.uk, and 2 hits there for Taysha and Neurogene on there. There's a clinical trials register in the EU. And then obviously the patient organisations as well will have information. Okay, so that's me, I think. Okay.